Okay, now we're going to talk about photoreception, which is the ability of an organism to detect light and generally also to tell something about that light. And I'm going to start at the most basic level and move up from there. So really we're going to start off with the molecules that are sensing that and move up from there. So really this all starts out with this molecule right here, which is 11-cis-retinol. And this is the molecule that is actually going to be interacting with the photons to cause the beginning of this reaction that is going to allow you and your brain to sense light. What happens is when a photon interacts with this molecule in the correct way, or the correct type of photon does, is going to change 11-cis-retinol into this, which is trans-retinol, or all-trans-retinol, and is going to make that conformational change and begin the whole process. So trans-retinol, or cis-retinol, whichever the case may be, is bound inside this protein. This protein is a class of proteins called opsins. Now, opsins are these transmembrane proteins, so this would be in a membrane area like this, and it has these 7 helical domains, transmembrane domains, that go through it, and that's where these opsins are. And these opsins then, this change in retinol from cis to the trans configuration of retinol is going to cause a change in opsin and cause opsin to become enzymatically active and start the rest of the chain reaction. So that's at the most basic level what's going on with photoreception. So let's talk a little bit about opsins. There's a couple different kinds of opsin. First off, there's rhodopsin. And if we look at across all organisms, rhodopsin is by far the most common of the opsins. And it responds to a very wide array of wavelengths. And so rhodopsin is going to respond to almost the entire visible spectrum, which is good if you only have one opsin. However, it's bad if you want to distinguish between different wavelengths of light. So rhodopsin does not really allow you to have color vision at all. If you just have rhodopsin, you don't have color vision. And the cells that have rhodopsin in them are not contributing to color vision either.
We also have these things called photopsins, which allow the discrimination of light beyond just presence or absence like rhodopsin does. Photopsins actually respond more maximally to different wavelengths of light. So we have red, green, and blue photopsins, and all of these, get that down there to blue, are what occur in humans. However, we also see some other photopsins in other places in the animal kingdom, such as some places or some organisms have ultraviolet photopsins. Some animals also have yellow photopsins. So photopsins are the types of opsins that allow for the discrimination of the wavelengths of light.
So the color of the opsins, and when I mean color of opsins, I'm not saying actually the physical color of the opsin if you were to pull out and look at it under the microscope. I'm talking about the color or the wavelengths of light that opsins respond to when I talk about colors of opsins. It actually turns out the retinol in all of these opsins is completely identical. There's no difference between the retinol between these opsins, so the actual molecule that's interacting with the photon. What turns out is that when you have different opsins, they interact with the retinol differently, and it's going to change the wavelengths that are optimally absorbed by retinol depending on the way that the opsin interacts with the retinol. So there's this kind of interaction back and forth that the opsins affect the retinol in terms of what wavelengths that retinol maximally absorbs, but then it also changes when the retinol changes from the cis to the trans conformation. It also changes the activity state of the opsins as well. So let's take a look at the photoreceptors that are going to be, or the receptors that are going to be detecting light. And they come in these 3 different main parts. What we have is called the outer segment. And the outer segment is made up of essentially one cilia. So oftentimes, or sometimes you'll hear it referred to that vertebrates have ciliary photoreceptors. It's because the main photoreceptive part of them is essentially this one cilia that has many folds in its membrane. So we have many of these membranous disc-like folds in this large membrane, or in the membrane of this large cilia that's out one end. And it's in this outer segment that we have opsins. So the opsins are bound to the membrane, and that's why we have all these folds in the membranes, to maximize the membrane to have these opsins inside of in this outer segment. Then what we have is this inner segment here. This inner segment is going to have all of essentially the cellular machinery. We have mitochondria in here, we have the nucleus in here, so this is where all of this is actually located and operated, and then we have the axonic terminal. And this is where it's going to interact with whatever other cell is passing off to. So whatever cell is going to be passing off its signal to is going to be going there. So actually all the color or all the light sensation is happening in this outer segment where the opsins are, in these many stacks of folded membranes in the cilia, and then we have kind of signal transduction and passing in the inner segment and axonic terminal. And that's kind of the basic makeup of a vertebrate ciliary photoreceptor. So let's talk about how this actually happens. What are the steps that happen in this reaction, in these reactions to transduce this signal? So first, a photon is going to hit retinol, and it's going to change retinol from the cis to the trans conformation. And then after it's changed from the cis to the trans conformation, the trans retinol is going to activate the opsin. So now the opsin is now enzymatically activated. And so opsin then, in turn, is going to activate another protein inside the cell. It's called transducin. And transducin, in turn, is going to activate photodiesterase, or phosphodiesterase, excuse me. So essentially what we have is a signal cascade here. Transducin then activating phosphodiesterase. Phosphodiesterase, in turn, is going to decrease the amount of cyclic GMP in the cell by breaking down the cyclic GMP. So now we're having less cyclic GMP. Well, it turns out in this outer segment we have cyclic GMP gated sodium channels. As we reduce the amount of cyclic GMP inside that cell, these sodium channels are going to close. And this is kind of the opposite of what we've been saying. Normally what we've seen is some stimuli is going to do something and it's going to lead to the opening of ion channels. And we're actually doing the exact opposite here. We are having some stimuli that is going to be closing ion channels. And this closing of ion channels is going to cause hyperpolarization. So we've been having these sodium channels let sodium in and essentially constantly depolarizing the cell, and since we close them, we get this hyperpolarization of the photoreceptor. We're going to come back to that. I'm going to leave it just for a moment. We're going to go back to talking about photoreceptors again just for a moment. We're going to come back to that. Keep that in mind. We have two different main types of photoreceptors. We have rods, and we call them rods because this outer segment, the cilia, looks like a rod, and that's what's on. And rods have rhodopsin. We also have cones. Cones are so shaped because their outer segment looks like a cone. They also have many fewer folds to the cilia membrane as well. So we have rods, we have cones.
So rods will contain only the opsin rhodopsin. And because they only contain rhodopsin, that means that rods are not going to be color sensitive. Instead, rods are just giving you really information about the luminance of light. So how bright is it? But isn't giving you any sort of color discrimination. However, cones, on the other hand, contain photopsins, And these cones are only going to contain one of the photopsins that we've talked about before, which means that these cones then are only going to respond to certain wavelengths of light. So for instance, we have long cones or L cones, so called because they respond to longer wavelengths of light, or red light. And therefore they're also sometimes called red photoreceptors. We also have medium cones or M cones, which respond to green wavelengths, or respond optimally to green wavelengths of light, therefore are going to be kind of giving you discrimination that those frequencies. And then we also have short or S cones, which tend to respond to blue light and will also be called sometimes blue photoreceptors. So therefore, because we have these three different kinds of cones that are responding more maximally to these three different wavelengths of light, because they contain these different opsins, and again, one cone is only going to contain one kind of opsin, we therefore can begin to discriminate light based on how much different cones are responding compared to the other ones.
So let's talk a little bit more about what's happening here on this side. We have some photon that's going to come in, strike this outer segment, cause the retinol in here to change from cis to trans and cause that cascade that we talked about earlier, is going to cause eventually sodium channels to close in this outer segment and cause a hyperpolarization. Well, it turns out that all the while before, we were having neurotransmitter released between this rod and this next cell, which is a bipolar neuron, or just a bipolar cell. This neurotransmitter was being released to this bipolar cell, but once we had the photon hit and the sodium channels close and we get this hyperpolarization, we are cutting out this neurotransmitter release. So essentially we are cutting off the signal from the photoreceptor. When it's exposed to light, it's stimulus. That might seem a little bit backwards, except the neurotransmitter that this photoreceptor is giving off is inhibitory. It's essentially putting on the brakes of this bipolar cell. So as we lift that inhibitory input from the photoreceptor, now we get a depolarization here in the bipolar cell. So we get a depolarization. Depolarization. Now, this is a graded depolarization. This is a graded potential that's going to happen in this bipolar cell because we're lifting the inhibitory influence of the photoreceptor once it's stimulated. We're going to cause this this depolarization that's going to cause this graded potential. It's going to release neurotransmitter onto this final cell, which is a ganglion cell. This ganglion cell then is going to have an axon that goes to the CNS, to the brain. It's not until this ganglion cell that we actually get an action potential or not. So that's where the graded potentials that we're getting from the photoreceptor cell, which is actually a hyperpolarization, which turns into a depolarization in the bipolar cell, which is then transmitted to this ganglion cell. It's not until here we actually determine, is this an action potential? Is this not an action potential? So that's essentially what's happening here. Inhibitory, this stimulus is inhibitory in the photoreceptor cell, or hyperpolarizing in the photoreceptor cell, which is inhibitory in the bipolar cell. So we're lifting off those brakes, which lets this bipolar cell depolarize and cause a depolarization in this ganglion cell, which essentially makes the decision, I use that rather anthropomorphically, makes a decision whether or not an action potential is going to happen or not. So that's essentially for each photoreceptor, the photoreceptive kind of conduction pathway that's going to happen with that receptor. So just to recap this visual pathway, a photoreceptor neurotransmitter, they inhibit the bipolar cell depolarization. And stimulation of this photoreceptor is going to lift its inhibition of the bipolar cells. Bipolar cells then have a great potential that they're going to transmit to these or the ganglion cells, which are then going to begin the first action potentials in this entire pathway. So let's talk a little bit about a few more comparisons between rods and cones. We talked a little bit about rods and cones, but it turns out that rods, they're actually quite a bit more sensitive to light. And here's why. First off, they have more pigments in their outer segments. You notice that we talked about earlier that there's a lot more folds in these outer segments, the cilia of rods. Their cilia tend to be larger as well. And so there's a lot more area there for photons to hit and cause a hyperpolarization in these rod cells. So that is one major reason why these rods are more sensitive to light than cones are. There's also a high amount of convergence in rod cells. So rod cells are going to be lifting inhibition on bipolar cells in a graded way. So greater stimulus, greater graded potential. And those bipolar cells are going to be giving this graded depolarization to ganglion. But it turns out that there's about 100 rods are converging onto each rod-associated ganglion cell, which means that you have a lot of convergence of information, but this allows for essentially amplification of that information. Each rod does not have to bring each ganglion cell to an action potential, to threshold by itself. We can actually have each rod only giving on average about 1/100th of the way to action potential, but if they're all doing that, we can bring the ganglion cell to threshold, given action potential, which means that any one rod with much less of a stimulus can still bring, along with its peers, other rod cells, a ganglion cell to action potential, which means that we don't need nearly as much light to activate the pathways that rods are associated with than we do with cones. And so this also has a consequence associated with it. Because we're reducing this information, because we're converging this information, we have a lot less acuity. Essentially, you can't distinguish. All these rods that are converging, you can't distinguish between them. You don't know if one rod cell in that pathway or another rod cell in that pathway actually had more or less light. You just know that all of them together got you an action potential in that ganglion. So because of this, you cannot, when you're using your rods more, essentially in low light, you do not have nearly the resolution that you have in highlight. Cone cells, on the other hand, are much more acute. Most cone cells are going to synapse onto one of their own, just their own ganglia. So we have one or fewer often, or excuse me, not one or fewer. We have one or only a few cones generally synapsing onto each cone ganglion cell, which means that all of that information is being kept separate. So we can distinguish between different cone cells, which means we have much greater resolution when we're talking about cones. Now this has the other problem. We need a lot more light to hit a cone cell and cause a lot larger graded potential in that cone cell to bring a ganglion cell to the action potential. But that's kind of a trade-off that we're getting. They can only be used in well-lit situations, but we get very good acuity out of them. We have a mix of these rods and cones in our eyes to give us the best kind of range of this, is that we have rods that work well in low light, cones that work in highlight and give us color vision as well. So we have this smaller receptive field for cones, so essentially the area, the spatial area that a cone cell can sense on, and this gives us greater acuity. Let's talk a little bit about light adaptation. So one major part of light adaptation is we're switching between cones and rods being predominantly used when we move from a dark situation to a light situation. You'll probably often notice that it takes your eyes a while to adjust. One of the reasons it takes a while for your eyes to adjust is that this cis-retinol complex and the opsin complex together, every time that's used, it needs to be regenerated. It can only be used once before that complex needs to be regenerated to be used again, and that takes time, which means when you're in highlight situations and the cis-retinol keeps getting turned into trans-retinol, it's taking time and your body's having a hard time keeping up, being able to regenerate this cis-retinol. Well, that's not actually such a bad thing, because it actually turns out that in highlight environments, cis-retinol gets depleted and is decreasing your sensitivity to light. It doesn't actually take very much depletion of the cis-retinol to decrease your light sensitivity. fact, if you only have a 0.6% reduction in your cis-retinol overall concentrations in your cells, this can actually result in a 3,000-fold decrease in light sensitivity. So even these minor depletions of cis-retinol in your eyes are going to lead to decreased light sensitivity, which is good in high light. If you walk from like a dark place out into a bright outdoors, you'll notice that's really hard to see for a while because you're being so overstimulated by the light. You need this reduction in sensitivity to get the best vision possible. And so this reduction isn't necessarily a bad thing. Well, it turns out the opposite is also occurring in darkness. As you go into darkness, you are having less light hit your photoreceptors and changing cis-retinol to transretinol, which means that your body has time to catch up in its regeneration of this transretinol or the cis-retinol. And as you catch up and build up your reserves of cis-retinol, your eyes become more sensitive to light. And so you get dark adaptation happening that way. This is also why this doesn't happen immediately. This isn't the only thing that's happening. Your pupils are also dilating or constricting. So this isn't the only method of light adaptation, but it's one of the major ones. This is one of the ones, and especially the one that's happening very slowly. That's taking several minutes to occur. Your pupils constricting, dilating, that happening very quickly, but this one is happening rather slowly. Okay, so now that you know basically on a cellular level how light is being transduced, let's talk about the makeup or the anatomy of the eye, so we can start looking a little bit more at what's happening. So here we have an eye, and we have two major compartments. We have this compartment here that is anterior to the lens, so this L right here, that is the lens. This is what's focusing the light. And what you have going on here is that in this compartment in front, we have a stuff called aqueous humor. This is a water-like fluid that is in front of here. In front of this, you have a cornea. Oh, let's go back to that. Cornea. And the cornea is going to be this clear layer that is going to protect your lens. And right here, we have our iris. The iris is made up of various muscles that are going to restrict this opening right here being your pupil. It's going to restrict how much light can get in here. We have this large chamber here, which is filled with another thicker liquid. That's called the vitreous humor. And then we have three major layers as we go through the eye. So the outermost layer here, this is the white of the eye, which is also known as the sclera. Inside here, we have this other layer that's carrying a lot of blood vessels, a lot of nutrition. This is the choroid membrane here. So the choroid is through there. And then here, this thin green layer that's right through here, this is actually the photosensitive area of the eye. This is the retina. So the retina is where our photoreceptors are going to be placed, and that is what is actually located right there. Coming out the back here is the accumulation of all the axons of all those ganglion cells that we had coming out of our photoreceptors, and this is the optic nerve. Okay, so let's take a look about how is this retina actually organized on here. So if we take a look at it, this is the retina. So what we have back here is the choroid. Up here we have the vitreous humor. And then if you go this way is actually also forward is the pupil. So this is where, this way, to the right. So we have light coming in here shown by these wavy arrows, coming in from the pupil through the vitreous humor. And the first thing it's going to interact with, or kind of have to pass by, is these ganglion cells. So it passes by the ganglion cells, passes by the bipolar cells, passes by the ends, the inner segments of these photoreceptors, and it's not till back here that we actually get to the photoreceptive part of those cells, the cilia. So what we have here up top here is we have some rods, we have a cone here. So it's not until back here that we actually get light interacting with the retinal, the opsins, and the cilia, the photoreceptive part of our photoreceptors. So this has a small consequence that you actually get some light refraction happening as it's passing through these other layers. Then we get this interaction happening back here, and then we get this signal transduced back up this way to the axonic terminals of the photoreceptors, to these bipolar cells to the ganglion cells, and then these ganglion cells to their axons that are kind of laid across the top of all this, across the top of the retina that this lights also happen to go through. So that is how the general arrangement of the cell happens, or of the retina is happening. And talking a little bit more about the anatomy of the retina, we have then, as a consequence of this, is axons and blood vessels that are lying over top of photoreceptors. And this is happening over most of the retina, except for one place, and that's at the fovea. The fovea is a small depression in essentially the center of your retina that has the highest concentration of cone cells, which means we have the highest acuity right there at the fovea. Also, all the axons and the blood vessels have been moved aside from the fovea. So actually the fovea does not have to take light that's been passing through axons and blood vessels like the rest of the retina does. It's getting a little bit more unobstructed pathway that light's coming to it. This fovea, however, is very small. It's only about the size of a pinhead, so it's very small. And one way that you can tell how small it is put your hand out in front of your face and look at the middle of the back of your hand. And you'll notice that you have some fine detail in the back of your hand. So look right in the center of the back of your hand. You notice that you have a lot of small folds there, a lot of contour, a lot of detail. We'll look right at the edge of the back of your hand. So now we're going from the center of the back of your hand. Look at the margin. Look at just alongside kind of the index finger. Keep it right there and try to notice how much of that detail of the back of your hand can you still notice. Really, it shouldn't be very much at all. Most of that detail has gone away. And when you think about it, if you think about how much of your visual field, how far that is away, that's not very far. This is a very small part of your visual field away from the very center of your vision, the fovea. So actually, you get a very noticeable decrease in your ability to notice detail, your acuity, outside this fovea. And that drops off really quickly, actually. So that's something I kind of keep in mind. Now, all these blood vessels and axons that are lying on top of all these photoreceptors have to get to the brain. They have to somehow get outside the eye. And they're exiting through the retina at the optic disc. And this is a place known also as your blind spot, where there's no photoreceptors, because we have all these axons they're needing to exit the retina and go towards the brain. And so there's really no space for any photoreceptors there. So you have this area of your retina that's also not receiving any light, this blind spot. And you really don't notice it. So we're going to finish up talking about the senses by spending a little bit of time with chemoreception. So chemoreception is just the detection of molecules in the environment, whether this is the internal environment or external environment or wherever it's coming from. So this can be kind of broken down into three basic places that we have chemoreception. The first one here is interoreception, so detecting the environment inside one's own body, and really that's going to be detecting things like oxygen, carbon dioxide, and other important molecules to maintain homeostasis inside the body. I'm not going to talk very much about interoreception, chemoreception right now, because we're going to come back to that a lot later on when we're talking about various other physiological principles, so we're just going to leave that one there for the moment. What we're really going to be talking about is these other two right here, taste and smell. So these are the two kind of most prominent, well-known applications of chemoreception in vertebrates. So let's start out with taste. So taste is detection of chemicals specifically in an aqueous medium. So our ability to taste it, in humans at least, is localized on our tongue. And the specific area of the tongue are in these things called taste buds that you've probably heard about before. Taste buds are going to be aggregations of these taste chemoreceptors. And just as a little bit of vocabulary here, a tastant, if I refer to a tastant from here on out, a tastant is a molecule that's going to stimulate the taste receptors. So that's what a tastant is. So, on your tongue you have these things called papilla like this. Papilla just being some sort of extension of a tissue or an organ or something like that. We have these papilla. You'll notice that your tongue is rough. It's not perfectly smooth. It's because we have these different papilla. And in between these papilla, we have these taste buds, like that. And so it's in between these different papilla that we actually have most of our taste buds, not directly on the surface of the tongue. And so if we zoom in specifically to one of these taste buds like this is kind of what you would see. So it's the actual most of the cells, or the chemoreceptor cells of the taste buds are kind of embedded down inside the tissue, and we have a couple different parts here that we want to talk about. First off is the opening into this, which is called the taste pore. And then we have a few other things. We have these long spindle-like cells that are the actual taste receptors. And then we have some other supporting cells as well. And so all of these come together to make these individual taste buds. And then back behind here, so lower down than that, we have the afferent nerves. Neurons. Say neurons here. They're going to synapse onto the lower end of these taste buds, or specifically the taste receptor cells, and carry that information to the brain. And at the top of these taste receptors, up in the taste pore, we have quite a few microvilli that are going to extend up into the taste pore and have many of the receptors on them specifically up there that are going to be interacting with the aqueous medium and whatever chemical composition is inside there, and then stimulating these taste receptors or not. So one interesting thing here is that actually taste receptors are just really modified epithelial cells that have a few special receptors on them and a few other things to be active. So let's talk a little bit about the specific taste pathways here. And really there are 5 known modes of taste. We have salty, sour, sweet, bitter, and umami, and those are the ones that we're going to be talking about. And the first ones that we're going to talk about are mediated by ion channels. The taste receptors are ion channels, so specifically salty taste is mediated by sodium channels that are in those microvilli at the top of the taste receptor. And these sodium channels are going to allow sodium to rush into the cell, cause a graded depolarization, or a graded potential, and that's the signal that you get for that. Sour, you have these proton channels, so just H+ channels that are going to allow protons into the cell, and again cause a depolarization of the membrane potential of these taste receptors. So sour, specifically what is detecting protons, is measuring essentially the pH of whatever, or the acidity of whatever it's sampling. So both of these are the simplest of the taste receptor molecules that we have, or excuse me, taste receptors that we have. The rest of them are G-coupled receptors. I'm not going to talk too awfully much about specifically how these G-coupled receptors are working. Just know that that is what's going on. So sweet is going to bind to small sugar molecules, as you probably know. Mono or disaccharides. And these G-coupled receptors, G-protein coupled receptors that we have for sweet, are going to then be using cyclic AMP or an IP3 second messenger system inside the cell. We have bitter. These are responding mostly to alkaline substances, although there's actually kind of a range of different substances that they're going to respond to, but most of these are going to be alkaline. And again, this is a G protein coupled receptor. And then also we have umami, which is essentially a Japanese word that means yummy, essentially. And this is going to be the savory flavors. And these are receptors specifically for amino acids. The one that is really prevalent here is glutamate. So you'll probably know that MSG, monosodium glutamate, is a popular or often used food additive, and is specifically made to interact with these umami taste receptors in your tongue and cause a savory sensation. Now, one thing I should point out here is that all 5 of these are present on most taste receptors, but you have certain taste receptor cells that tend to have mostly one or the other, but most of these are on all those taste receptor cells. Okay, so let's move on from taste and talk a little bit about smell, or olfaction. So olfaction is just the detection of chemicals in the air. And really, olfaction is using a very different strategy than taste is, whereas taste is sampling specific whole molecules out of the aqueous environment. Olfaction, instead of detecting the entire molecule, has receptors for specific molecule components of these molecules, so like an aldehyde group or some other sorts of group or some aspect of the molecule, rather than looking at the entire molecule. And so the sensation of smell is then constructed out of these responses to these different components. So let's kind of give you an illustration of how this would work. So imagine we just have these 5 different receptors up here for olfaction. That's not really the case. In fact, humans have thousands of different receptors in their olfactory pathways. But let's just imagine we had only these 5. And imagine we have certain odorants. Again, an odorant, like a tastant, is just some molecule that is going to be stimulating the olfactory receptors. And so imagine we're looking at this first one up here. We notice that it has 2 components. It has both this sharp side and it has this rounded side back here. So it would bind effectively to this first receptor and the second receptor, the 2 highlighted red, but would not respond to these next 3. Next, this other one, this kind of pentagon-looking shape over here, again has a pointy end to it, so it would interact with this receptor, not with the rounded one because it has no rounded shape, but with this kind of 3-sided end here. It would interact with that, but not the other 2. So in this way, by just sampling the different pieces of this, we can use 5 different receptors, but we can differentiate many more than 5 different molecules because we're just sensing aspects of them, and because we can get these different combinations of receptors being active, it means that we can get a much higher variety of sensation out of these receptors than just the number of receptors that we have. And so this is kind of the different strategy that olfaction is taking rather than taste is taking. So let's take a really quick look at what's going on here with the sense of smell. Here you have your nose. Probably don't need to be told where your nose is. And here are your nostrils right here, and some air is going to be coming in this way. And it's going to be interacting, the molecules in there, with this area right up here, which is the olfactory mucosa, which is the area in which sense of smell is taking place. Right above the olfactory mucosa, right here, is this specific part of the brain that's lying just beyond this small piece of bone right here. And right above that is this area right here, which is the olfactory bulb. And so the olfactory bulb is actually where the initial processing of these senses of smell are going to be taking place. So if we look at the magnified view of this, again we have our olfactory bulb here. And then down below we have this area, which this is actually bone right here, that's separating the nasal passages from the central nervous system, the brain up here. And then we have our receptors down here. So let's go ahead and clear all that off here just for a second and look a little bit more at this. So down here in the olfactory mucosa, what you're seeing is these blue, red, and green olfactory receptors. And these old olfactory receptors. And the interesting thing about these olfactory receptors, unlike a lot of the other senses that we've been looking at, in which you have a specialized receptor cell that's not really a neuron, some sort of receptor cell, that is then going to be excited by the stimuli and then pass off this information to a neuron on the other side of that, some sort of afferent neuron. The olfactory receptors are actually specialized neurons that have these cilia at the very ends, where the dendrites would normally be, that have these olfactory receptors on them. Also in this area, we'll have different types of cells as well in the olfactory mucosa. Alongside these olfactory receptors, we also have support cells. S-U-P-P-O-R-T, support cells. And these support cells are going to be creating this mucus layer that is covering up the ciliated ends of these olfactory receptor neurons. So those are support cells. Also back here, kind of towards in here, we would also have another kind of cell sitting out back here, and these are going to be basal cells. And these basal cells will actually turn into olfactory receptors later on. So your olfactory receptors are actually being turned over fairly rapidly, and these basal cells will become new olfactory receptors, which is actually very interesting because these olfactory receptors, not only are they afferent neurons that have this sensory end on one end, they actually, their axons extend all the way into the brain and take the information straight into there. And these neurons, despite having a sensory end and another end that goes into the brain, are being turned over fairly commonly. Okay, so let's look at what's going on back behind here now. So you'll notice that all of these different types of receptors segregate themselves out into their different types, and they are going to converge in these areas called the glomeruli. And these are areas in which many of these receptors are going to be synapsing onto these other neurons inside the CNS, which are the mitral cells. So what's happening here is essentially you're getting all these different receptors types that are being segregated out into the different types and then converging on or synapsing on specific central nervous system neurons specific for that kind of information. So this is where in this olfactory bulb here is we're having the very first kind of relay station that is directing the different kind of scent information to the correct places of the central nervous system. So really that's where I'm going to kind of wrap it up. There's more that we could talk about the sense of smell, but I'm not going to go incredibly into that. And we're going to come back to chemoreception later on in the quarter as we're talking about a lot more places with interoreception and the specific types of chemoreception going on there. But for now, we're going to leave the senses with that and go ahead and move on to muscle physiology after this.